Look around. Do you think you're all alone? Completely wrong. You cannot see them, but there are lots of bacteria, and not just around, they're also on you, and some of them can cause disease. Usually, we fight them with antibiotics, but abusing of these drugs has led to another problem: multi-resistance bacteria, also known as superbugs. Not all of them, but some bacteria can acquire resistance to one or more antibiotics by transfer their genetic material from one another. Many multi-resistance bacterial strains are appearing, and the pharmaceutical industry cannot follow the pace. Let's talk about phages. A phage is a virus that hosts only bacteria. Many phages are composed of a head or capsid and a tail. The head is a protein shell, which keeps the genome and viral proteins necessary for infection. The tail enables attachment to specific structures on the bacteria's surface, what makes phages specific infectors. Once attached, the virus inserts its genetic information inside the bacteria, and within minutes, bacterial machinery is forced to synthesize copies of the virus. There are two different strategies to infect. There is the lytic cycle, which consists in destroying the host right after copies are ready to infect new cells. And there is also the lysogenic cycle, in which the virus stays dormant, passing their genomic information to the progeny of the infected bacteria. These dormant viruses can be activated and follow the same process as in the lytic cycle. Phage therapy consists of the use of phage to infect the bacteria that cause disease. This presents various advantages. Specificity. They only recognize a narrow range of receptors, even just one bacterial strain, what implies minimal impact on our normal bacterial flora. Humans lack these receptors, so we won't be infected. Autodosing. Phages use their host to replicate, so if there is bacterial infection, they will multiply in an exponential way. Coevolution. Bacteria can also develop resistance to phages, but these have the property of mutating easily, so it's faster to find new phage strains that can overcome this resistance. Drug development is much slower. Solution to exotoxins. Some bacteria produce toxins highly harmful to us. By destroying the bacteria, all these toxins are released to our blood, producing what is called a septic shock. A solution could be administering phages with lysogenic cycle that block toxin synthesis and do not destroy immediately the cell. There are also disadvantages. Narrow action range. Because of the high specificity of phages, in a case of acute infection where the bacteria is unknown, treatment with a single phage would not be enough. We would have to administrate a cocktail of phages. Immune response. Phages are, after all, viruses, so there is a possibility that our immune system recognizes their capsid proteins and creates an immune response. This problem could be solved by hiding these proteins behind other molecules. Not all bacteria. Some bacteria infect our cells and stay inside. Therefore, phages cannot detect and destroy them. Nowadays, phages are widely used in the food industry to reduce bacterial food infections, as well as controlling contamination of drinking water. Nevertheless, Western countries haven't yet moved forward to phage therapy in humans, although it has been used for almost 100 years in countries that belonged to the old Soviet Union. Should now be the moment to consider as allies what we have always seen as an enemy?